Hello! This lab aimed to analyze different plant samples for various microbes and see how they may or may not help the organism survive in heavy metal contaminated environments. From these samples, certain microbes were found to be plant beneficial in protecting the organisms from heavy metals. The samples analyzed in this lab were taken from a Superfund site in Saraland, Alabama, Red Wing Carriers Incorporated, site number 13. 2 samples were taken from this site and analyzed for their epiphytes to find nanoparticle synthesizing abilities. Sample 1 was Potamogeton crispus, a curly-leaf pondweed. Sample 2 was Hedera helix, a common ivy. Plants were not able to tolerate heavy metal contamination for various reasons, but microbes can offer unique benefits so that plants can defend against heavy metal toxicity. Heavy metals are metals with high densities and atomic masses/numbers that are toxic at low concentrations. Some examples of heavy metals are mercury, cadmium, arsenic, chromium, thallium, and lead. Some of the effects of heavy metals on plants are that they trigger oxidative stress, which can cause the plant to become necrotic. The plant can develop chlorosis, which hurts its ability to photosynthesize, and it also changes various biochemical pathways in the plant. Certain microbes can produce nanoparticles that can help aid the plant in reducing the negative effects that come with such contamination by bioreducing metal ions. This can be done through processes such as methylation, demethylation, metal oxidation, intra- and extracellular metal sequestration, and many other ways. First, the plant samples were collected from Site 13, and then microbes found with them were isolated and screened. Sample 1 was Potamogeton crispus, a curly-leaf pondweed. Sample 2 was Hedera helix, a common ivy. Growth media was made to grow the microbes. The growth media was a lysogeny media made of 40 grams to the liter of LB broth and 1 liter of water. The exact quantities used in this lab differed as different numbers of agar medium were made depending on the quantity needed. About 300 milliliters of the LB broth was made per group. Then the mixture was autoclaved to sterilize it. Once this step was complete, the liquid was poured into petri dishes. Then the dishes were streaked with microbes that were taken from the samples. After this, the next step was to incubate the bacteria in tubes that contained nitrate broth and then centrifuging it to separate the supernatant and the biomass from one another. Then, 1 to 2 drops of 0.8 solution of sulfonilic acid and alpha-naphtholamine solution were mixed. This tested to see if the bacterial colonies had nitrate reductase activity of the isolates, showing if they possessed the ability to produce silver nanoparticles. If a pink color appeared, then the test was positive. The samples that tested positive were VQ1, 2, 3, 4, 7, and 9. A second test was done to test for silver reductase activity. This was done by adding 1 mm of silver nitrate. If a dark brown color formed, this meant that silver nanoparticles formed. The samples that tested positive were the same, VQ1, 2, 3, 4, 7, and 9. Next, a PCR reaction was done. To do this, a lysis buffer was made with the bacterial culture and then neutralized with tris-HCl. This is beneficial to denature the DNA and detach it from the histones. It also stabilizes the pH, which is necessary in order to amplify the genes. After the lysis buffer is done, the tube is centrifuged and the 16S RNA amplification can be further processed. The 16S gene is used because it is optimal length and it contains all the necessary information. It is also easily amplified by PCR, the next step. A primer must be selected for the PCR reaction. The best primer has the following: 18 to 24 base pairs in length, about 40 to 60% nucleotide GC content. It must start and end with 1 or 2 nucleotide GC pairs. It needs melting temperature at about 50 to 60 degrees Celsius. The primer pairs should be within 5 degrees of their melting temperatures, and they should not have complementary regions. Next, the PCR reaction is done. This happens in 3 steps. First, the double-stranded DNA is denatured at about 94 degrees Celsius where the strands separate. Then it's annealed at about 50 degrees Celsius where the primer attaches, and then it is extended at 72 degrees Celsius. Next, gel electrophoresis was done in agarose gel. Agarose gel is used because it does not produce electroendosmosis. It only needs about 2 milliliters of sample. It does not bind the protein, and it does not affect migration of the DNA. Once the gel was loaded, the gel was run at about 80 to 150 volts until the dye line was about 3/4 of the way down the gel. Next, the PCR reaction was cleaned up to avoid any remaining DNTPs and primers from interfering with the experiment. The DNA concentration was analyzed to see the absorbance measures. Sanger sequencing was used for this step. After all DNA sequences were collected, they were run on a program called DNA Subway. The BLAST technology found the best match for each sample according to their DNA sequences. The results are as follows. Samples VQ1, 2, 3, 4, 7, and 9 tested positive for nitrase reductase activity. The same samples tested positive for silver reductase as well. After the samples were processed through BLAST, they were identified. VQ1 did not match anything in the database. VQ2 matched Bacillus thiogenesis. VQ3 matched Arthrobacter oriscens. VQ4 matched Pseudomonas grammis. VQ7, renamed VQ5 in the Excel sheet, matched Arthrobacter oriscens. VQ9, renamed VQ6 in the Excel sheet, matched Pseudomonas lutea. The hypothesis for this research project aimed to see if nanoparticles could be made from the microbes to aid the plants in defending themselves from toxic heavy metals. Bacillus thiogenesis showed that it helped plants in growth promoting and biological control. Although there is no direct studies done on this specific strain of Arthrobacter oriscens, studies have shown that the plasmids of Arthrobacter help the plant with heavy metal resistance. This bacterial type, Arthrobacter, is found in many Superfund sites around the globe. Again, there are no direct studies done on the specific strains of Pseudomonas grammis or lutea. Studies have found that Pseudomonas as a group aid in heavy metal remediation. They can produce metal chelating cytiophores and surface-active extracellular polymeric substances. Metal chelating cytiophores help remove certain metals from the soil around the plant so that it can live longer. Surface-active extracellular polymeric substances aid in lessening the effects of heavy metals on plants. All samples that were identified are all plant beneficial. This research supports the hypothesis, confirming that these microbes do produce nanoparticles that help plants defend from heavy metal toxicity. In conclusion, this research supports the hypothesis, confirming that these microbes do produce nanoparticles that help defend from heavy metal toxicity. Thank you, Banup Muhan, for leading an amazing class this semester and allowing me not only to learn about genetics, but see it in the real world. Thank you, Dr. Mukhtar, for teaching an amazing class and truly wanting your students to excel. I really appreciated my time in this class. Shown here are the references used in the making of this presentation. Thank you for listening.